I'm Thiesli Sente, and I will be presenting a novel epitope-based peptide vaccine against the Zaire Ebolavirus, designed through Immunoinformatics. So there are two things I want to establish about the disease. Number one, the virus is severely lethal, so its mortality rate peaked at 89%, killing more than half of the infected. So number two, it is also extremely contagious. In fact, it is the most virulent strain of the five that are known to exist. So regarding the transmission of the virus in severely infected patients, less than 1% are at risk when you haven't been exposed to the sick patient. 2% when you live in the same household, 23% when you've had direct exposure already, and 53% when you are the caretaker of the patient. So finally, 83% of those who are handling the corpses of those who were sick become at risk for getting the infection. So these are people who get rid of the body or perform burial rites. The structure of the Ebola virus comprises of 7 genes, but out of the 7, the glycoprotein is what is usually selected in the making of antiviral compounds and vaccines. So the glycoprotein is also the only protein that is expressed on the surface of the virus, appearing as spikes. So this protein is involved in the entry of a virus onto a host cell through attachment and membrane fusion. So additionally, the Ebola virus also has a strong binding affinity to antigen presenting cells, which in turn leads to massive recruitments of other dendritic cells and macrophages. So targeting the glycoprotein can inhibit viral entry and membrane fusion, as well as induce the innate and acquired immune system because of its high binding affinity to MHC class molecules. So in this study, three types of epitopes are going to be the bulk of the vaccine. So CTL stands for cytotoxic T lymphocytes. So their job is to induce apoptosis, which means cell death of the infected cells. HTL, on the other hand, helper T lymphocytes, they initiate the adaptive immunity by releasing cytokines after activation. So this leads to differentiation of B lymphocytes and other effector cells. Lastly, we have our B lymphocytes, so they can retain memory and are the ones who secrete antibodies after differentiation to plasma cells. So a vaccine that targets all three kinds of epitopes, can strongly boost the immunogenicity of the vaccine. So the objectives are as follows: to predict, assess, and filter the CTL, HTL, and LBL epitopes of the vaccine. Number two, to select the epitope and HLA combinations predicted to be recognized by West Africa and the whole world. And number three, to employ an appropriate adjuvant and specific linkers for the vaccine assembly. So there are 5 strains of Ebola viruses. However, this study will only compile and discuss the data of the Zaire subtype. So among the 7 genes present in the structure of the Ebola virus, only the sequence of the glycoprotein will be acquired and evaluated for the prediction of epitopes. So this study will also only use human models as the host for the virus. So it will not cater to other animal models such as non-human primates. So this study will also not cover the production, manufacturing, and delivery systems of the vaccine. So NCBI will be used for retrieving the FOSTA sequence of the targeted protein, and then after that, NetCTL will be the server used for CTL prediction. So the assessment is done by checking first the immunogenicity, toxicity, allergenicity, and finally antigenicity. So epitopes that pass these parameters are then partnered with MHC1 alleles. For HTL prediction, it uses the IEDB server first and simultaneously partners it already with MHC2 alleles. So after that, the same parameters will be used for filtering in addition to cytokine inducibility. In LBL prediction, the same parameters follows also. So this is just an example of what the procedure and assessment and filtering would look like. Multiple sequence alignment will be done to account for mutations, and population coverage of the CTL and HTL epitopes is done to determine the global coverage of the vaccine, partnered with MHC alleles, and the assembling of the vaccine will include selecting an adjuvant and linkers to string the epitopes together. The assessment of the fully put together vaccine will also will be tested again using the same parameters in addition to physicochemical properties, which are identified to validate the stability of the sequence. So after that, the in silico simulation is then done to see the real-life immunogenicity profile of the vaccine. So what you want to see here is how long it will last, how high the activity is, and how protective the vaccine will be in a span of 1 year. So table 1 shows the accession number and other details regarding the FASTA sequence of the Zaire Ebolavirus glycoprotein. So all succeeding experiments will come from this sequence. All peptide sequences that are displayed here are arranged by immunogenicity in descending order and have been filtered out to be immunogenic, non-toxic, non-allergenic, and are antigenic above a zero point 0.8 threshold. So initially I had 213 CTL epitopes, but through filtering I was left with only 10 candidate epitopes. All allele partnering has been filtered out to have a percentile rank score of 1 or below, since a low percentile rank score suggests high binding affinity to the CTL epitopes, which is what we want to have a successful antigen presentation. IEDB was able to predict 51 combinations under a 0.25 percentile rank threshold, and then after filtering I was able to obtain only one peptide that was able to induce all three kinds of cytokines. These are IFN gamma, IL-4, and IL-10. So the allelic partnering of this one peptide was also shown to have a sequence of 4 combinations. So in LBL prediction, initially I had 174 epitopes that were predicted, but after filtering, the table shows that I was only left with 5 candidate LBL epitopes. The figures on display here are the results of the multiple sequence alignment viewed with the MU server. So as you can see, very minimal mutations were identified, and the epitopes in this study are highlighted in yellow. I was able to count how many conserved epitopes there were in the 533 random sequences aligned. So almost all similarity scores exceed 96%. Tapos, this means that the epitopes would be able to induce CTLs, HTLs, and LBLs regardless of where the infected may catch the virus from. So this means the chosen epitopes would allow for a widely distributed vaccine in terms of strain mutation, making the design of the construct persistent and efficacious. In population coverage, globally the vaccine is predicted to cover 100%. So this means that the epitope and HLA combinations of the vaccine are predicted to be recognized by the global population. As you can see here, West Africa also had a score of 99.18%. The adjuvant used was CD40L, which is a co-stimulatory molecule involved in the activation of helper T cells and B cells. So since it's involved in the success of antigen presentation, that was the one that I picked. So EAAK linker was a spacer used to connect the adjuvant to CTL. Tapos AAY was used in between CTLs. GPGPG was used to connect the CTL to the HTL. And to connect HTL to LBLs, we used the spacer KK. And KK was also used in between LBLs. The properties of the now strain construct include the retention of its antigenicity, allergenicity, and toxicity. So physicochemical properties also imply the stability and solubility of the vaccine. So without giving too much away, here you can see that the exposure of the antigens present in the vaccine was able to induce the immune responses of CTLs. For the immunogenic profile of HTL, for helper T lymphocytes, their population was shown to be very present throughout the year, even after exposure from the vaccine. So a graph of the cytokines released shows the cytokines IFN gamma, which is involved in viral interference, had the highest peaks between the days of injection. And for the graph of LBL, the graphs displayed shows the population of B cells, plasma B cells, and the antibodies secreted during the exposure of the vaccine. So the vaccine proves to be long-lasting since active B cells were seen to be in abundance throughout the rest of the year.